And once again, I'd like to welcome everyone to the Los Angeles Birders webinar for tonight. We have a wonderful webinar coming up, and to introduce that webinar, we have Tori Fay, our program chair. Tori.
Thanks so much, Ron. Good night, or good not good night. Hello, good night. There we go. Let's start with that. I'm not saying goodbye yet. Tonight we are pleased to welcome Joan Ferrer Obiol, a self-described seabird addict, outdoors lover, and evolutionary biologist whose work focuses on how bird populations diverge and how those evolutionary units may respond to climate change. At the young age of 9, Joan already had a passion for birds and bird migration. He began research in high school with a project on the Mediterranean gull and later earned his undergrad degree from the University of Barcelona, or I should say Barcelona. Upon graduation, he spent a year conducting fieldwork on seabirds with the French Polar Institute on the sub-Antarctic Kerguelen Islands, which piqued his curiosity about how wide-ranging species like seabirds speciate. After that, he returned to the University of Barcelona to complete his master's and PhD degrees in evolutionary and population genomics. Joan is currently a postdoctoral research with the Bird Genoscape Project at Colorado State University. This large international research effort uses genomic data to map patterns of population structure and connectivity across the entire ranges of bird species. Specifically, Joan's research brings together genomic information with more familiar lines of evidence, such as morphology, ecology, and geography, to help clarify species boundaries and improve our ability to anticipate how bird populations may respond to threats like habitat loss and climate change. So please join me in welcoming Joan Ferrer Obiol. And I'm
Thank you,
going to go ahead and
Tori,
stop sharing my screen. You're welcome, Joan.
for this really nice introduction. So I will share my screen, I guess. Here it is. And we'll do highlights. Editing. Okay, so good evening, everybody. As Tori said, I'm Joan Ferrer, and today I want to talk to you about how we can harness genomic data, what that means data about the genome of species, to delineate species and conservation units and predict their responses to climate change. So I'll give first a little more personal introduction. Tori did an amazing job. I think she basically went through all the introduction I wanted to do, but well. So I'm, as you can tell probably from my accent, not from this country. I'm from Spain, specifically from a little, well, close to a little city called Tarragona, which is a Roman city in the area of Catalonia, which is this northeast area of Spain where we speak another language that is Catalan. And in my area, we are also like a bit crazy. We enjoy doing these things that you can see in the picture, which are called human towers, which is basically us climbing on top of each other. This is one of my favorite pastimes when I'm back at home, and I used to be like an active member of one of those groups, but unfortunately, no human tower groups in Colorado where I'm at the moment. Um, as I'm a geneticist as well, I did a little experiment on myself. I wanted to sequence my genome and kind of see if the area where I'm born is also actually quite representative of my ancestry. It turns out that boringly, it kind of is. So I am 97.7% Spanish and specifically Catalan from my region. But I still have like a 1.4% Western Asian North African, which I'm quite proud of. So as Tori also said, I was bitten by the bird back like quite early on, as you can see in this picture from me being when I was 9, assessing mold limits in this chief chaff. But my interest in birds started really with seabirds. Seabirds were my first love for birds, and specifically the Mediterranean gull, which in my region, close to Tarragona, we get like 75, well, 7 to 75% of the world population of this seabird. I also love, and more recently, I've actually transitioned more towards proper seabirds like Procellariforms, And here you can see me very happy with this Cape Verde shearwater, which was a rarity that we found in a colony of quarry shearwaters that I used to work in the Canary Islands. So as Tori also said, I studied an undergrad in biology at the University of Barcelona. And when I finished my studies in biology, I didn't really know what I wanted to do. I knew I wanted to work with birds, but I didn't actually know specifically what. So I actually had this amazing opportunity to go to the subantarctic islands of Kerguelen, which are, as you can see in this map, in the middle of nowhere between South Africa, Australia, and Antarctica. And they are a seabird paradise. These islands hold some of the biggest king penguin colonies in the world. They have colonies of lots of different albatross species and lots of other seabirds. And there I was in charge of a French science program where I was taking samples of seabirds and sea mammals, participating in long-term surveys, equipping different animals with loggers. I love this as like a little fun story. So these loggers that we used to put on the people put still on elephant seals are amazing. And actually, most of the information we have nowadays about the water column in Antarctica comes from these devices. Male elephant seals can go down to 3,000 meters depth, which is insane, like much more than any submarines that we've ever built. So I was there for a long time. I was there for 14 months. As you can see, like that was time enough to see weird things like this four-winged giant petrel. And that gave me time to think about what did I want to do. And then I was really interested in demography and speciation as well. But like I didn't know anything about genetics. And when I was there, a Canadian researcher told me that using genomics, you could study the evolutionary history of species and look at demography, for example, across the whole history of the species, which I found amazing. So when I came back from Kerguelen, I went back to Barcelona, studied a master's in genetics and genomics, which was a bit tough after all that, but that was a great opportunity and it allowed me to pursue with a PhD on seabird genomics. After that, I changed my city, Barcelona, with a nice cathedral for another city with a nice cathedral. That's Milan, where I did a postdoc working on more seabird species and also some land bird species that I will be talking to you about today. After that, I crossed the Atlantic like a vagrant and I went to Colorado to work on the bird genoscape project, which I will also briefly talk to you about today. So that's enough about me. So for a little outline of the talk today, so I'm going to start presenting some work from my PhD. Specifically, I will talk to you about a study I did on the species delimitation of North Atlantic and Mediterranean shearwaters. After that, I will talk to you about one of my main projects from my postdoc in Milan, which was on the lesser kestrel and defining conservation units and assessing their responses to climate change. And finally, I will talk to you a little bit about what do we do within the bird genoscape project and how we use genomics to aid bird conservation. So as a little introduction, since the publication of The Origin of Species by Darwin, the idea that species share a common ancestor has become a fundamental idea in evolutionary biology. So for example, in birds, from a common ancestor that lived alongside Archaeopteryx around 150 million years, birds have evolved into a myriad of shapes, forms, colors, and they have colonized all the ecosystems on Earth. They are some of the most successful vertebrates on the planet. But all those species, how did they arise? So how did all these species arise from this common ancestor, right? That's through speciation or the generation of new species, which is a continuous process that is completed with the attainment of reproductive isolation between two different evolutionary lineages. However, this is, as I say, a continuous process that is not deterministic, and it can actually go back at any moment. So doing what we call a speciation reversal. So if this speciation is a continuous process, we might expect that variation in nature will be also continuous, right? Like we won't really see species. We will not be able to like define species because variation will always be continuous. But that's not true. Variation in nature is generally not continuous. And that's generally due to two sorts of processes. Like there's adaptive processes, like the example here with the birds of paradise, that through what we call sexual selection, they've evolved into this myriad of like beautiful and different plumages. And then there's also neutral processes that can bring to variation in nature, phenotypic variation in this case, like this case of the leaf warblers, which is not as spectacular, but it also generates variation. However, despite this like continuous pattern of speciation, biodiversity conservation relies on the delimitation of species and conservation units to be able to protect our the species, right? So that raises the question of like, so what is a species, right? And that is not a debate, that's a really hot debate, and it's not a debate I'm gonna tackle here, but it's important to talk about one concept of species, which was described by D.K. Rose in 2007, which is the general lineage concept. Under this species concept, species can be considered as separately evolving metapopulation lineages or distinct branches in a phylogenetic tree. And the cool thing of this species concept is that it puts together all the previous species concepts that were there, and it focuses on their commonalities, which makes makes it possible to use science-based methods to delimit species. However, we know that speciation is actually attained with the attainment of reproductive isolation. So reproductive isolation is clearly an important criteria for species delimitation, right? And some authors actually argue that we need to take this into account. And to be able to take this into account, the best way is to use an integrative approach to species delimitation using other sources of data apart from genetic data, such as phenotypic, like morphological data, also ecological data, and that can actually tell us if the species are reproductively isolated or not. But focusing on species as the unit for conservation can actually be counterproductive, because within species, we have genetic variation and adaptive potential that differs between different parts of the species breeding range, or the species range, basically. So more recently, some authors have advocated for the use of conservation units, which are intraspecific groupings that are delineated with the goal of maximizing a species' evolutionary potential and can be used to pinpoint areas across species range that should be prioritized for protection, right? And in this case, they don't need to be reproductively isolated. They just need to be differentiated at the genetic and ecological level. There are different ways, and there's lots of different ways of defining these conservation units, but two of the most popular ones are evolutionary significant units and adaptive units. Evolutionary significant units are actually populations or group of populations that are characterized by high genetic and ecological distinctiveness. While adaptive units, which is a similar concept, but in this case they are focused on groups that differ in their adaptive variation. So in genetic variation that is actually linked to either local adaptation or like that is actually of an adaptive nature. So to put this into practice, let's delve into the example about species delimitation in North Atlantic and Mediterranean shearwaters. This study focused on a group of morphologically quite similar species but two different sizes within the group. That's like from one side is the medium-sized Balearic, Yelko one, and Manx shearwaters on the left, and on the other hand, there's the small shearwaters like the Sargasso voids and Barolo shearwaters. So resolving the phylogenetic relationship, so like the species tree of shearwaters, has been very challenging. And so that was one of the objectives of my PhD. Originally, people thought that species that were small were actually related to each other, while species that were of a more middle-sized were also more related to each other. But using genomic data during my PhD, we resolved that the group of North Atlantic and Mediterranean shearwaters, which comprises both small and medium-sized shearwaters, is actually a monophyletic group. That means that they have a common ancestor among themselves. And the cool thing as well that we found is that the evolution of small and medium body sizes has occurred several times throughout the species tree of shearwaters. So then we focused on this group of North Atlantic and Mediterranean shearwaters, and we sampled across all the species and subspecies across all their range to try and use this genomic data to assess their their species delimitation, right? So first of all, we looked at a PCA, which is this plot here that basically shows the genetic variation reduced in 2 dimensions, okay? So the different clades here represent, in this case, different groups of species. So we saw basically with this PCA approach, we saw 4 different groups, a Mediterranean group in green on the bottom, then a Manx shearwater group on the top in orange, and 2 groups for the little shearwaters, the Sargasso shearwater and the Boyds and Barolo shearwater. And using another dimension, we even saw that Barolo and Boyd shearwater also constitute different groups. So these groups that we saw with this approximation actually match some of the species that had been defined in different bird lists. But as you can see, the different bird lists that had been proposed were differed quite a lot in the number of species that of shearwater of North Atlantic and Mediterranean shearwaters that they considered, between 3 and 6 recognized species. So here, using our genomic data, we actually used several methods using like machine learning techniques and also what we call as multi-species coalescent approaches to see how many clades, how many potential species did we see. And we came out with a criteria that like if more than half of the methods agreed, we would, and then we had also evidence from other sources supporting species status, we would define species. And if less than half of these methods agreed and there was like signature of genetic differentiation, but not very strong, we would actually define those as conservation units. So using this approach, we actually came out with a species delimitation proposal, which included 5 species, the Manx shearwater, the Mediterranean shearwater that included both the Balearic and Yelkoan shearwater, the Sargasso shearwater, and the Boyds and Barolo shearwaters. And we also defined 4 additional conservation units, the Manx shearwater from the northeast of the distribution, and the Canary Island bank shearwater, and from the other side, the Mediterranean shearwater, like Yelkoan and Balearic shearwater. And we also highlighted the need for more data for Sargasso shearwater because there might be 2 conservation units there, but our sampling was not exhaustive enough to be able to look at that. Okay, so after this first example, let's switch gears and talk about this project that I was involved during my postdoc at the University of Milan with this amazing species that is the Lessa kestrel. So the Lessa kestrel is a species, is a beautiful raptor, colonial, that is a specialist of open areas in hot and dry climates across Eurasia. And it is a migratory species that performs a trans-Saharan migration towards sub-Saharan Africa and the south of Africa. Unfortunately, the species is of conservation concern, especially in Europe, but potentially also in Asia, although it's less evaluated there. And that's due to a steep population decline in the second half of the 20th century, mostly due to habitat loss and the use of pesticides. And also the dry period, an extended dry period in the Sahel. So in our study, what we wanted to look at was, first of all, we wanted to infer intraspecific evolutionary lineages or conservation units, as we defined them before, and assess their ecological differentiation to see if they are actual conservation units. Then we also wanted to understand if lesser seem to be adapted to their local environmental conditions. And finally, we wanted to give historical context by investigating demographic and distributional responses to past climatic fluctuations to basically give us support to be able to assess the lineage-specific vulnerability to future climate change. So we started with a big network of collaborators that allowed us to get genetic sampling from across the whole range. That was we had like more than 30 collaborators from all across different, like 20 different countries. And then we also curated a big data set of breeding and non-breeding occurrence records from eBird, from country breeding censuses, and lots of other sources of data. So using the genetic data, we first did study of genetic differentiation, we wanted to assess the genetic variation across the range. And we actually detected low levels of population structure despite the strong habitat fragmentation in the species. But we found two main genetic clusters, one in the west that can be seen here in orange, and one in the east that can be seen in this plot in blue. And interestingly, the individuals from the Middle East show kind of like mixed ancestry, suggesting migration or gene flow, as we call it, from the eastern to the western lineage. We then wanted to assess if these two genetic clusters were also ecologically differentiated. And first, what we did is use this big data set of occurrence records to model, using species distribution models, the distribution at present of the two different clades. And during the breeding season, we saw that there was very little overlap between the suitable area of the eastern group and the western group. And they actually matched the known distribution for the species really well, providing support for this model, right? And we also saw that there was very little overlap between the non-breeding season distribution of these two genetic groups. So they seem to actually target different areas. And within these areas, we also saw that they have marked differences in the habitat use and selection. So while lesser kestrels rely primarily on herbaceous vegetation, birds from the western lineage actually rely on croplands quite a lot, especially during the breeding season. And also during the breeding season, especially, they rely on human-modified environments, and especially in urban environments, to breed. So yeah, it seems that the western lineage shows a stronger association with human-modified environments than the eastern one. We also evaluated if the climatic niches of these species were different. So this is a complicated plot, but we don't need to go very much in detail. You can just focus on these little blurbs, or like the little kernels, of two different colors, which represent the two different genetic groups. And these two axes here represent temperature on the x-axis and precipitation in the y-axis. And as you can see, there's very little overlap between the climatic niches of both lineages. And the Western lineage seems to occupy warmer areas year-round and more humid areas during the breeding season. So we have two genetically and ecologically differentiated lineages. So next we wanted to know, okay, these two different genetic groups seem to occupy two very different two very different landscapes, right? But can we actually show that there's genetic signatures of them being adapted to these different environments? To do that, we did what we call a genotype environment association analysis, which is a type of analysis that looks for genes or like genetic loci that are associated with particular climatic variables across the range of a species. So using that, we found 60 genes that are associated with the environment. And we found that these genes are actually much more associated with the environment than the rest of the genes. And that's what we see in this plot as the bold blue and red lines compared to the black lines. And we also saw that the frequency of these loci is very different between Western and Eastern lineages. So that basically tells us that lesser kestrels, yes, they seem to be adapted to different environmental conditions and to their local environmental conditions. So next, we wanted to be able to hindcast the effects of past climatic changes on lesser kestrel populations. And to do that, we inferred population sizes using the genomic data. That's a fascinating thing that we can do. Using genetic data, we can actually trace population size of a species across time, sometimes with just as much as one single individual. And a cool thing that we discovered, we also traced the suitable area across time. And we basically saw that effective population size and range size co-vary with climatic shifts, decreasing from the last interglacial to the last glacial maximum as temperatures got colder and as is a warm adapted species that's expected, and increasing from the last glacial maximum to the mid-Holocene. Really, like another cool thing that we found is that the divergence history between the different lineages also was affected by climatic shifts. Actually, the divergence time between Western and Eastern lineages coincided with a decrease in effective population size and a range contraction during the last glacial period. And then the admixture and like the expansion of the range expansion also coincided with an increase in population size in the Western ESU especially. So lesser kestrels have been affected by past climatic fluctuations. So these actually gave us a very strong evidence that these species seems to be responding to climatic fluctuations. So then we wanted to understand if we, given that, we can forecast the effects of ongoing climate change on these lesser kestrel populations. And to do that, we did different analyses. One of them was to actually extrapolate the results from the species distribution models towards the future. And a really cool thing that cool and concerning thing that we found is that geographic isolation between the Western and the Eastern lineages is expected to increase in the future, which could end up with like promoting genetic differentiation and ultimately speciation. We also saw that we expect range expansions for the Western lineage, while we expect range contractions for the Eastern lineage. And this is true for both the breeding and the non-breeding distribution. And these results actually are quite in agreement, are really in agreement with the results from the recent trends during the last 10,000 years, but actually due to the impact of climate change is expected to actually change even more abruptly. Another thing that we looked at is that Eastern populations that already perform longer migrations are expected to increase even more these migratory distances in the near future, unless they find a new area to winter, right? And finally, another thing to assess the vulnerability to climate change that we looked at is, so remember when I said before, those analyses that we did called the genotype environment association analysis, which looked for genes that are associated with the environment. So we can use those genes to look at what's the relationship between the allele frequencies of those genes and different environmental conditions at present day to be able to forecast how these allele frequencies would have to change in the future to adapt. And in areas where these allele frequencies should change more abruptly are areas that are potentially more under threat for climate change, because that's quite difficult to do in a short period of time. And here again, we saw that the Eastern conservation unit seemed to have a higher risk, of a higher vulnerability towards climate maladaptation. So taking all these together, we can say that these evolutionary distinct lineages actually show divergent responses to climate change. So after this example, I would like to switch gears and talk to you about the Bird Genoscape project. I won't talk to you about specifically about what I'm doing within the Bird Genoscape project, because this is still in early stages and would have to be for another talk in the future, if you guys invite me again. But the Bird Genoscape, but I will kind of give you an overview of what do we do within the Bird Genoscape project to use genomics to help bird conservation. So the main aim of the Bird Genoscape project is to map population level migratory pathways. And we do that using genomics. And then we use those population connectivities to aid conservation. And I will go through a few examples of how, of some conservation applications of what we do. And finally, I will also talk a little bit about some outreach and education that we do within the Bird Genoscape project. So migratory species have quite a complex life history, like annual cycle, which normally consists in the breeding period, in the breeding range, where the species tend to breed and reproduce. That breeding period is generally followed by fall migration to go towards generally lower latitudes to spend a stationary non-breeding period, which is generally just being able to like feed and get energy back to migrate again during the spring migration towards again the breeding grounds. So that's generally like how migratory species life, like annual cycle looks like. So why is mapping connections across the full life cycle important to avian conservation? So, well, we know that from recent studies that North American avifauna has been declining strongly. We've actually lost nearly 3 billion birds since the 1970s. And this is actually due to stresses that occur across the breeding, migratory, and wintering areas of the species. And these stresses can be like predation, especially by alien species like cats, habitat destruction, disease, or climate change. So connecting populations across the full annual cycle allows us to understand how stresses at one particular stage of the life cycle are connected to impacts at another stage. But so this is great, right? But connecting populations is not an easy task. How do we do that, right? So you probably are all familiar with one of the first approaches that we use to be able to identify migratory connections, which is bird banding. Bird banding is a great resource and it has actually allowed us to know a lot about bird migration, but it has the main issue that recaptures tend to be rare. So we need a huge volume of individuals banded to be able to get enough recaptures to understand how different populations migrate. Another more recent way of studying bird migration is through radar detection systems. And this is really cool. Most of you probably are familiar with the birdcast. But the thing about this is that it actually is really useful to understand how migration depends and how migration differs depending on meteorological conditions, for example, but it does not allow us to track individuals or like even population level migration at all, because it's just at the level of all birds, right? Then another more like quite popular way, maybe the most popular way of tracking migratory connections is through GPS and radio tags. And that's actually technology that allows us to have a very precise connection between the different areas that a species visit during their life cycle. But the problem of these devices is that they tend to be expensive and they also tend to, it's difficult to put them on lots of individuals. And they also are actually quite difficult to, like, sometimes you have to recover them to be able to download the data. So what's our approach to establish these connections? So we use feathers and we use genomics to connect these populations. So how are we able to actually sequence their genomes with feather samples? So that's thanks to the fact that the tissue at the base of a feather contains DNA. And this DNA, we can isolate it and we can sequence it. And this allows us to find unique genetic code that allows us to trace the breeding origins of migrants captured anywhere along their migratory trajectory. So to do that, the Bird Genoscape project during the last years has actually been able to connect and like has had the help of a lot of NGOs, government, academic people, and museum partners as well. This is a map, for example, of all of some of the breeding and wintering samples that we have at the moment. So thanks to all these collaborators, we get around 10 to 20,000 samples every year. Now the full collection of the Bird Genoscape project is about 260,000 feathers of more than 100 species and across a time series of more than 25 years. So it's an incredible resource that we have thanks to all these collaborators. And of course, like curating all these samples is not easy task. So how do we do sample curation in the lab? That's thanks to like an amazing team of Colorado State University undergrads. Here you can see some of them that work on sample intake and associating BGP IDs to every sample that comes to the lab. Then these samples, we generally group them by species prior to storage. And our lab manager, Amanda, you can see her really happy because she has all the samples neatly organized and put into the freezers. So once we have these samples, what do we do with them? Generally, to generate what we call a genoscape or a map of genetic variation, what we do is to sequence around 175 to 100 genomes across the breeding range of the species. That's done by our lab manager, Amanda Carpenter. And Amanda does the isolation of DNA, prepares the genomic libraries, and sends them for sequencing. Then we receive the sequencing data, and here you can see our bioinformatician, Kristen Bossu, um, processing the genomic data using bioinformatics. And once this genomic data is processed, we can build those maps of genetic variation across the range, what we call a genoscape. So here you can see an example for the American redstart, where each of these colors is a different genetic unit. Then what we can do is to sequence genomes of wintering birds, and using different software that has been developed in the lab, we can assign those individuals back to their breeding colonies. So how can we incorporate information on migratory connections into conservation efforts, right? Like once we get these migratory connections, as we explained, how can we use that? So we start with these genoscapes, as we said, plus the wintering locations and the wintering assignments. And that allows us to build or like to model the connection strength between the breeding and the wintering sites. That's what's called migratory connectivity. And migratory connectivity is stronger when distinct breeding populations are concentrated in similar areas on the wintering grounds, and is very weak when breeding populations intermix all together and winter in similar wintering locations. So here, for example, you can see an example of how we can combine this connectivity data across different taxa to ask questions such as which wintering regions support the highest number of distinct genetic units, right? And here you can see a map showing the number of distinct breeding units per all these ecoregions. And in this case, we have like preliminary result that shows that lowland Central America, at least for the species we assessed in here, that were assessed in here, shows the highest number of distinct breeding units. But we might say that maybe that for conservation is not of great use because not all of these units will actually be of conservation concern, right? So one thing we can do is also look at which wintering regions actually exhibit the highest proportion of vulnerable populations. And that we can do it using metrics like the R2R, which is like combines different measures of vulnerability. Then we can set a threshold for like highly vulnerable populations, and we can map them in their wintering areas. And in this case, we see that this map is quite different. And in this case, it's highland interior Mexico and northwestern Mexico, which have the highest proportion of vulnerable populations. So that's for the science, but what are we doing beyond the science within the Bird Genoscape project? So one cool thing I wanted to show you is that in the recent years, the Bird Genoscape project established a partnership with the National Audubon Society to be able to see if we can use the genoscapes to improve the visualizations within the Migratory Bird Explorer website. So basically, what we managed to do with help of them, like collaborating with Audubon, is to replace these maps that seem quite static, right? Of like where the breeding grounds and the wintering grounds seem like a single unit, with these maps that are much cooler, which show the different genetic units and that show as well the migratory connections between the breeding and wintering areas. So yeah, I would like to thank a lot of people that are written in here for the different projects that I showcase you today. And I would really like to thank you all for your attention. And I'm really happy to take any questions.
Wonderful. That was wonderful. If you have any questions, please pop them into the Q&As button, I guess, down at the bottom of your screen. That helps us kind of keep track of everything. And I thought it was a wonderful, wonderful talk. Let me change the view here. But you did lose me already. It lost me at the four-winged petrel. I wasn't sure what to make. But but I did have a question. You talked also about, a little bit about speciation reversal.
Yeah.
And do you have any examples of that?
Yeah. So we've worked with the shear waters with a good example. Actually, the in the Mediterranean, we have two species of Puffinus shear waters, which I showed in there, which are the Balearic and the Yelkovan shear waters, which basically have disjunct breeding areas, right?
Mm-hmm.
So originally, researchers looked at the mitochondrial DNA, and these two groups seem to be very differentiated. And that's what actually brought them, that together with like some morphological differences,
Oh,
to propose that those two were different species. But then using genomic data across the whole genome, we realized that they did not differ that much. They had some differences, but nothing that was like fixed between the species, right?
right.
So a master student that worked with us actually did a project on looking at this in detail. And what we actually understood is that what had happened with these two clades was that during glacial periods, they had speciated. But
Mm-hmm.
then during interglacial periods, they came into contact again. And given that there was still not strong reproductive isolation, they were able to continue breeding among themselves and basically remove any differences that they had during that
Interesting.
time. And that has happened over and over during the quaternary period,
Those freezes
glacial
and
and interglacial cycles.
interesting. And that's through the fossil record to be able to see.
So through a fossil record, we can see, but that's just through genomics, actually. Like
Oh,
with the genomic data, we can like look at the demographic history and we can see how they have changed in population size during these times.
interesting. Interesting.
I see Lily has a question in the chat. How did you find out the kestrel populations changed during the Ice Age?
So we found that out using two different methods. Like one of them was looking at the genomes and doing what we call demographic inference analysis. And those are these type of analysis that allow us to understand, thanks to how the genetic variation is, it allows us to know at each time period in the past how the population has changed. And then like using what we call like a mutation rate for the species, right? If we know how many changes in the DNA they accumulate with time, we are able to calibrate those changes to a specific time in the
Mm,
past, right? And then we also used what we call demographic modeling, which allows us to model the history of divergence and gene flow between the different units or the different evolutionary lineages within a species. And that's also like, but all thanks to the genomic data.
interesting. Thank you.
Thank you.
Thank you.
I actually had
I've
a
got a couple
question.
questions.
Oh,
Oh, sorry, go ahead, Calvin.
thanks, Tori. I had a quick question. I was wondering about the when you're talking about the Genoscape project, if you had any more examples of like conservation takeaways from the Genoscape project so far, whether that's for specific species that you've been able to learn something about based on the migratory pathways, or just the broader like conservation projects in the like really densely vulnerable areas.
Mhm.
If you could speak about that at all.
Yeah. So one of the projects I'm involved, like for my work now in the Virginoscape is to actually like extend these comparative studies to 40 different species for which we already have range-wide data. And one of our aims is to actually do vulnerability maps thanks to the genetic data, but also based on other factors and basically put all these maps together for the different species to try and pinpoint areas that might harbor a lot of conservation units that are endangered, for example, right? And that would be a great resource to be able to understand which areas require prioritization for conservation. Another example now is that like another colleague and some people in the lab are working on a grassland bird project, because grassland birds are the birds that are actually experiencing the biggest declines during the last years. So that's together with the Bird Conservancy of the Rockies, and which have like really detailed monitoring of lots of grassland species, and thus they know what are the recent trends at a lot of like a huge part of the range of different species. And putting together the genetic, and now like the people in the lab are putting together the genetic information with all these other data on the monitoring to be able to actually pinpoint as well areas that might be the best for promoting conservation there.
Awesome, thank you.
So many of us are birders, and I think it's really, fascinating to think that these birds that we're just having to watch in our, our local patches near us have these very different histories about where they spend different parts of the year. That's we've had a fair number of webinars that kind of poked into this topic. And I'm particularly interested when you talked about the kestrels and how the kestrel, you have those sort of two different pathways that the kestrels, two different populations, and how they're diverging. And so I just think it's really fascinating to see how you're using the genetics to sort of like see their pathways and see what's happening. And I was really interested in how you see that one part of the eastern kestrel population is kind of somewhat crashing, and the western one seems to be doing better, or at least have the potential to be doing better. And I guess I have 1,000 questions, but I guess the main question I'm curious about right now is I'm wondering if you tried to pair that together at all with current observations, like maybe looking at eBird data, to see if there's any evidence of those things actually starting to happen with both those Western and Eastern populations. I'm really interested to see your collaboration with the Audubon Society and to see how you're updating those maps. And I'm wondering if like there's a way to make use of all these sort of citizen science sort of databases to help see if these kinds of things are happening. And then I have a totally unrelated question, which is I'm curious about how AI might be changing the Genoscape project. So off to you.
Great. Wow, those are great questions, all of them. So for the first question, yes, like that was one of the things that, because of course, like all the work I presented here for the lesser kestrels, this is all based on what we know now in the present. We try and extrapolate it either back in the past using genomics, or towards the future using also genomics plus species distribution models, right? And the climate projections that we have. So of course, there's a huge element of uncertainty that we try to like include in it by using different models of climate projections. But still, like we can't test this hypothesis, right? Like the only way we can do it is at least like seeing if there's already some trends. And actually, we saw that there clearly is a trend. So South Africa, which is where the eastern population of lesser kestrels spends their winter, the population has been crashing since the last 10 years. And they've seen that since they have like detailed wintering atlases for South Africa. So unfortunately, we don't have a lot of information for the breeding grounds because there's not a lot of work being done there, but we can see that on the wintering grounds. And on the other hand, we also see that in the western one, we see new colonies being formed in northern latitudes every year. So this group is clearly expanding. In this case, it's funny because for the first one, we have evidence for the wintering. For the second, we have evidence for the breeding, and we don't have much evidence for the wintering. But yeah, like these things seem to like go in agreement with what we had found. Then you asked me about, sorry, I forgot the second one.
About AI.
Yeah, AI. Yeah. So clearly, like there's some people that have already developed some methods that use what we call convolutional neural networks, which is a type of machine learning, to try and actually get to where a particular genetic sample comes from, right? And so there are methods that some people claim that they are working well already. we tried some of them for the genoscapes. Well, I didn't try them per se, but like the team tried, and they did not work as well as the methods we have at the moment. But there's for sure room for new methods that use machine learning to actually, yeah, like improve. One of the things that we can improve, and that it's kind of like in the process at the moment, is that for the moment it's very easy for us to assign individuals to a particular genetic population, but we can't really assign things on a more continuous way, right? Like within this particular genetic group, where in particular this individual might come from. And that's something that AI might help at, but there's other ways. Yeah, there's lots of different methods that are being developed for that.
Yeah, yeah,
Great,
that's
thank you.
interesting. Lance, one of our board members, asks, which species are you currently working on and why?
So I'm working on, as I said, 40 different species, and that's mostly like North American songbirds. It's all North American birds, but it's mostly songbirds and also a few woodpecker species, the burrowing owl, and we're trying to include also some other species into the data set. But basically, the species that I'm working on, some of them is species that the Bird Genoscape project had already worked on, and so it's data that we already had. Then there's all these new grassland species that we've been working on recently, due to the fact that, yeah, grassland species seem to be the ones declining the most during the last years. Then we also have a big data set of boreal distributed species, and that's because we have a collaboration with Ben Winger's lab. And he's been really interested in, and I think you had a webinar by Teresa Pegan a while ago, that was actually showing that data set. So I'm working on the species that Teresa worked as well. So basically, it's a mix of like which species do we get funding to work with, based on this, like the grassland birds, for example, and what are like other opportunities we have through collaborators or etc. And sample and species for which we have enough samples that have been shipped.
Interesting, interesting. Yvonne has a question about the last map in your presentation. She was wondering if you could show and explain that map.
Yeah. So let's again. So I guess she refers to that one, right? So here's where we show that like we go from this map that the Migratory Bird Explorer has for most of the species to this map. So here what you can see is that the breeding range now has different colors. And these different colors actually correspond to the different genetic groups that we had found by doing the genoscape. So it's basically a representation of this genoscape. And when you see that there's areas like this area here, where there's not a strong assignment to a particular group, that means that populations there are probably kind of like a mixture between different genetic populations. Okay. And that's why they are not clearly assigned to a particular breeding population. And then the cool thing in here is as well that like within the wintering area, we can include data on the individuals that we sampled genetics, like genetically, and we can actually show for each of the dots from which population they come from. So that gives an idea like we would have with GPSs of what's the connection between breeding and wintering sites.
Interesting, interesting. I may be mispronouncing this, but is it known, Yvonne asks, is it known what the genotype environment associated loci code for the kestrel population you studied?
Yeah.
Okay.
Yeah, so you were asking, sorry, that if that's the...
Is it known what the genotype environment associated loci code for the kestrel population? Well, do you know
Yeah,
this code for
yeah.
the
So
yeah?
exactly. So the genes that we found that were associated with the environment were actually all of them had been found previously in different vertebrate species, mostly
Ah,
birds, but we also all the vertebrate species, as being genes that are involved in local adaptation. That
oh,
means that are very differentiated between different parts of the range that are actually in very different habitats, right? So yeah, so that's actually a good way for us to see that these associations are not just spurious, but actually match on genes that we know that have functions that are, and that can be functions such as fatty acid deposition, for example, how it allows them to deposit fatty acids before migration, easier or less easy, right?
right.
So yeah, we found genes like that.
Interesting, interesting. Um, and our last question kind of loops back around to our first question. You got to explain that four-winged petrel.
So I'll put it again so as you can see it with more detail because it's such a cool one. So
It just
basically that yeah,
yeah, there we are.
yeah, there you are.
It's like a
Yeah.
helicopter petrol. This
Exactly. So that was like a really fortunate picture where it seems that there's only two legs there, and it seems that there's four wings. But
yeah,
basically, it was a very fortunate picture, as you can see. It's not
yeah.
modified. I didn't modify it at all. It's just
And
a picture.
no
And yeah,
Photoshop involved.
no Photoshop involved. No.
That is wonderful. Well, thank you very, very much for a wonderful presentation. We really enjoyed it.
Thank you
Uh,
guys for inviting me. It was really great. Yeah,
well,
I really
good. Thank
enjoyed
you.
it.
Thank you very much. And I hope everyone can join us next month. We have some wonderful webinars coming up. In fact, we have wonderful webinars coming up already planned through the first half of 2026. Thank you, Tori.
Right.
And we have some wonderful events. Birding 101 is around the corner. If you can and want to go to Costa Rica, this is your opportunity. And with that, good night, everyone. Thank you all for joining
Thank you,
us.
Joan.
Thank
Thank
you.
you.
Thank you very much, Juan.
Bye. Thank
Good
you.
night, everyone.
Good night.